Alrighty, here we go again. So, assuming that you have read the Google Classroom announcement, yes, I have the flu, so I will be back as soon as I can, Thursday at the earliest. So, we're going to do another round of notes like this today. We're going to do another set tomorrow, so be sure to bring in your headphones for both. What we're going to do really quickly before we go into translation, which is the second part after transcription, we're going to quickly review mitosis. Next, you're going to do some group practice with protein synthesis. You're going to work in small groups. You can work with 3 to 4 people. And then you're going to spend the rest of the time working on your translation homework. So what you can do right now, you can pause. You can answer these 4 questions. Keep your catalyst sheet until I come back. So don't give it into the sub. Don't put it in the basket because there's not going to be anyone to actually return it to you. So go ahead, pause, copy these things down, and then we'll move on. So coming back, I just wanted to quickly review the cell cycle and mitosis. Just a quick reminder that we have G1, S, G2, and finally the M phase when we're talking about the cell cycle. G1, just like you guys know, this is called gap 1. Think the cells are growing, they're making new proteins, they're making new organelles. The S phase, that is for DNA synthesis, when it's doing DNA replication, so when you go from one strand of DNA to 2 strands. The G2 phase is the very last step. It's preparing for actual mitosis when we're splitting the cell. If you haven't figured this out yet, this is review from notes number 19. This is not the new set of notes. And then finally, all of these things together, G1, S, and G2, we call all of these things interphase. So interphase as a whole, you can see it's most of the time here. That is where the cell is spending most of its time. It's getting ready to go. And then finally, this M phase is broken down into 2 different parts. So mitosis is when the nucleus splits in 2, so 1 to 2 nuclei. And then cytokinesis is when the cytoplasm and cell membrane divide. So it's like a karate chop right down the middle. So it separates those 2 cells entirely. So we go from 1 to 2 cells. And I looked over briefly some of your quiz scores. I think people struggled with kind of explaining what was going on for each of the stages in PMAT. So we're going to go through them. But we can break down mitosis down even further into those 4 phases. Before we get into mitosis, you have interphase. That's when it's in chromatin form. So G1, S, and G2. In prophase, this is when we go from chromatin to chromosomes. That's when it goes from the loose stringy DNA into the nice coiled X's. And the nuclear envelope also breaks down. A lot of people forgot that. The nuclear envelope has to break down so the chromosomes can go to opposite ends. Next we have metaphase, think M for middle. The chromosomes are going to line up in the middle and the spindle fibers are going to attach. So the spindle fibers are the little ropes that are going to pull them apart. Speaking of apart, in anaphase, that's when the sister chromatids, the 2 little arms, are splitting right down the middle, and they're going to go to opposite ends of the cell. And then finally, in telophase, this is when the chromosomes go back to chromatin, and the nucleus starts to reform. And then finally, cytokinesis is when we actually split that cell into 2. So up at the top here, you can see telophase, where we have 2 separate nuclei. They have completely formed. And then cytokinesis, when that actually splits down the middle, cuts it right down the middle, and we have 2 separate cells. That is back in chromatin form now. But now we are going to move on to notes number 21, talking about translation. So you guys talked yesterday about the central dogma of biology, which is DNA is converted to RNA, which is then converted into protein. Yesterday you talked about transcription, so it's when we go from DNA to RNA, and today we're going to talk about translation, where we go from RNA to protein. The way that I like to think about this is DNA to RNA, you're still in nucleotides. You're just rewriting. That's what transcription means. Script is to write. RNA to protein, we're going from nucleotides to amino acids. So we actually have to translate that message. So like if I were to translate this page into German, it would go into another language. That's what happens in translation. We go from nucleic acids to amino acids. So the genetic code, we call these the language of the mRNA. And the important thing when we talk about this is a codon. So it is a 3-letter word that makes a particular amino acid. So for example, these 3 nucleotides would code for leucine, which is a specific amino acid. These 3 code for valine. You don't have to memorize these, I'm just telling you what they are. These 3 code for another amino acid. These 3 code for another amino acid. And these 3 code for another amino acid. Notice that each one of these is 3 nucleotides. So a codon is 3. And each of these 3 nucleotides codes for 1 amino acid. It could be different, it could be the same, and we'll see how we figure that out. So translation happens at the ribosome. And this is when we go from our mRNA, our messenger RNA, to a polypeptide, so a protein. We're putting those amino acids together, we are making a protein. So this happens in a couple of different steps. Step 1, the mRNA, it leaves a nucleus after transcription and it goes to a ribosome. So the messenger RNA has reached a ribosome. That's this little pink kind of squarish thing right here. So it is attached. Step 2, each tRNA or transfer RNA carries an amino acid to the complementary sequence of the mRNA. So just like we talked about yesterday, U goes with A now, but everything else is the same. So notice that these tRNA molecules, they match perfectly. They're complementary. They have to be able to match like a puzzle piece so that it can bring in the correct protein, the correct amino acid. Step 3, the ribosome forms bonds between the amino acids to form a polypeptide chain. So you can see that right here. This amino acid has bonded to this amino acid, has bonded to this amino acid, so on and so forth until it gets the final polypeptide chain. And in step 4, once that tRNA brings in its specific amino acid, it'll float away. It'll grab another amino acid to be ready to bring into another ribosome spot. So feel free to rewatch this if you need some more review. But the way that you might have seen this before in like a benchmark test or something, now you actually will understand how it's used. But like we said, DNA is converted to mRNA, and with that mRNA, we can then determine what amino acid will be added on. And we use this codon chart to show what amino acids are produced from the codons. And you can see there's 3 amino acids right here. You're also going to see this on your homework page as well as the bottom of your notes. So if you need to reference that, you can look at either one of those. So for example, I want you guys to look at your mRNA codon chart and look for these certain amino acids. I'm going to give you guys a little bit of time. But what you're going to do, I'm going to show you back here. So for example, UCC. What you would do, you go over here, okay, I'm looking for something that starts with a U. And then I need to find something that has a C next and a C. So your amino acid for this one should be serine. So let's see, UCC is serine. So they're going to have either 3-letter codes or a full name. Don't worry about memorizing these, but just know that they are shortened. So I'm going to let you guys search for AGA, UGU, and UGA. Go ahead and pause if you need more time. But other than that, you should have gotten arginine, cysteine, and a stop codon. So stop codon means it has no amino acid. That's where the amino acid chain is going to stop. That's the end of the polypeptide. And the way this would work just from the beginning, so let's say you had a strand of DNA right here. You had your DNA, TTA, CG, your mRNA, you're going to do the complementary basis. So T would go with what, A would go with what, so on and so forth. To give you this, T goes with A now goes with U in mRNA, C with G with C, T with A with U again, T with A, C with G. What you're going to see is it's going to say, okay, what are our codons? And like we said, codon is 3 amino acids. And what you're going to do is just kind of break them apart so you can see them better. So we have our codons right here. And what you would then do is look these up in your amino acid chart, in your mRNA codon chart, to give you that amino acid. So that's asparagine, alanine, and a stop codon. So that's how that works. So you have your DNA, you do transcription to find your mRNA. Oops, sorry, that was my dog. And then it's going to do translation to find the following amino acids. So this one, it's telling you to go backwards, and this is your first question in your translation practice that you're going to work with partners. So in this case, it gave you the mRNA. So you're going to have to work backwards to find this. So just like we've seen before, you're going to find your complementary bases. It would give you something like this. And like we saw at the one point in the notes, the tRNA has to be complementary to the mRNA, so it matches up like a puzzle piece. So in this case, A goes with U. I'll show you. A goes with U goes with A, G goes with C, so on and so forth. And I'll show you a shortcut right here. But the DNA and the tRNA should be the exact same sequence, except for instead of a T in DNA, you have a U here. So it'll be exactly the same, except anywhere that you had a T, it is now a U instead. And to look up your amino acids, what you're going to do is break your mRNA up into your 3 nucleotide codons, just like this, so on and so forth. And you're going to look that up in your chart. So if you did that, if you broke these all down, oops, I don't have a codon chart in front of me right now, but I believe that you'll be able to find it. So you're going to look up AUG, and then ACU, and then AGC, UGG, GGG, UAU, UAC, UUU, and then UAG. So that's what you will use to find your amino acids. Don't let this confuse you. You will always, always, always use your mRNA. If this was a little confusing, feel free to rewatch this video. I will put up an Amoeba Sisters review if you want to watch that, or send me any email with your questions. But other than that, what you're working on, group practice with your protein synthesis. You can work in small groups for that. Work on your homework, and then work on any missing work that you might have. You can study with the Quizlet Live in the Google Classroom. And you can also still work on your science fair project. But other than that, I will hopefully see you on Thursday, if not on Friday. Please be good, and I will see you as soon as I can. All right, bye-bye.